I didn't.
Okay, let's start the class. First of all, next Thursday, we will have a lab midterm exam. So the lecture we will not have, because I want you fully prepared. And the first lab section will start 11 o'clock in the lab room and then followed by 12:30, 2, and 3:30. So I give you very fair 11 o'clock to start the exam, and don't think about the first lab is going to be due too early and the last lab is too late. So the first lab you don't have to get up too early. You can take your time, get up 9 o'clock, have a piece of bread, have a cup of coffee, and then you can review, and then you go to the classroom. So it's enough time, okay? So that's very... And of course, September 27th in the lab, we'll do the review. We'll do the roughly review for the lab exam. Okay. For the lecture, I put already is the after class reading, and you should definitely read it. This is basically is what the material what I put in the slides put on the blackboard, which is the details of things what we're gonna read. So lots of stuff here, and lots of the examples for the case study, and we will talk about that later. So here you see the conclusion: Staphylococcus aureus, epidermis, saprophyticus, pyogenes, all these things. Okay, so in Staphylococcus, we have a case study which is for you. Some of these case studies I will directly move to the exam paper. I'm seeing whether you studied or not. Okay, this is what I concluded from the lots of the textbooks. I did it like 5 years ago, so you should definitely read these. And also in our lab menu, there is also some material I need to print it out to give to you. Some of them are similar, but some of them are new. So you see, Corynebacteria, I wrote that paragraph let you read, and the Klebsiella case study, Haemophilus influenza, all these things. So you should definitely read. Okay, that's very important. Now let's talk about today's lecture section. We're not going to jump into here right now. We will talk about is some of the review. So like I said, we finished about the Streptococcus and the Staphylococcus. So we're going to first of all do a review, because otherwise it's too much information. So we need to refresh your mind to do the review. Okay, so first of all, what we are talking about here is gram-positive cocci. And we mentioned is Streptococcus and Staphylococcus. So I just write strep and staph. So the first thing first you need to know, they are all the same, gram-positive cocci. Streptococcus is basically is a chain arrangement, and the Staphylococcus is grape shape. That is from the gram standpoint. And now we mentioned there is a one biochemistry reaction can differentiate that very easy, is Catalase Test. And the Catalase Test, you always remember, Streptococcus is negative, Staphylococcus is positive, which means it's bubbling. Okay? So this is the basic thing. Then we talk about each of these one by one. Streptococcus, we talk about is alpha hemolytic and beta hemolytic. And of course, we mentioned about gamma hemolytic. This is all down on blood agar. So all right here, all down on blood agar. So blood agar, we will do real quick in the lab, is a differentiated medium. So alpha hemolytic, basically we talk about these two bacteria, is Streptococcus pneumoniae. Another one is Viridine Streptococcus. This is actually a group of Streptococcus, including Bovis, Mitis, mutans lot. Beta hemolytic, we are talking about is Streptococcus pyogenes and Streptococcus agalactia. And this is we call GBS and the upper one we call GAS. Now, since they're all alpha hemolytic and beta hemolytic, how we differentiate that? Remember, differentiate here we using is we call a P-disc. And the P-disc, we separate them into two. Streptococcus pneumoniae is actually susceptible, so we write S. And the Viridine streptococcus is resistant. And the major components of a P-disc is we call it opticam. Opticam. Okay, then beta hemolytic, we have a pyogenes and agalactia. How we differentiate that? We use A disc. Very similar. Streptococcus pyogenes is susceptible, and Streptococcus agalactia is resistant. And what is the components of the A disc? Is bacitracin. And then we mentioned about here is GAS, GBS. This comes out with a concept called Rebecca Lansfield group. So we end up with GAS, GBS. We also have a GDS, group D strep. What is that? That is Enterococcus facilis. Facilis. And this guy is basically, we call it resistant to 6.5% salt, and could change S-culin become S-culating, so it is a black colony. It's a black color. Okay? So this is our conclusion right here. Then let's move this. That's for Streptococcus. Okay, Staphylococcus. We talk about it's a grape shape. It is Catalase Test positive. It's bubbling. So what are we talking about the Staphylococcus? We talk about Staphylococcus aureus. Sorry, let's just talk about being in order. Staphylococcus epidermis. We talk about Staphylococcus saprophyticus. We talk about the Staphylococcus aureus. Now, we wanted to also talk about all these things. First of all, is on blood agar. This is non-hemolytic, we call it a gamma hemolytic. Staphylococcus saprophyticus is also gamma hemolytic. And the Staphylococcus aureus is beta hemolytic. And we will do in the lab, let's see whether it is. The second one is a mannitol salt agar. We will do real quick in the lab. Mannitol salt agar. And what we will find, the Staphylococcus aureus is yellow color. And epidermis, saprophyticus, both are colorless. Because those mannitol fermentation negative, and the last one is mannitol fermentation positive. Okay, now we end up with noble bouncing. Low concentration of noble bouncing. And we have a joke. Staphylococcus saprophyticus, we call it a honeymoon bacterium. And why we say it? Because we can testing it's resistant to the low concentration of noble bouncing. So this is resistant. The other two are susceptible. And last thing we talk about is Coagulase test. Coagulase test, which means gluten or coagulation of the blood cell, Staphylococcus aureus is positive, both of these are negative. Okay, so this is a conclusion table. Talk about all these from the bacterial media or biochemistry standpoint, how we differentiate that, the Streptococcus and the Staphylococcus. So we did the overall conclusion for all these things before we're going to move on to today's class. Okay, so take notes and the study, and also this is some conclusion from the after class reading material what I put on the eCampus. Okay, so let's move on to today's lecture. We're going to talk about is human disease caused by bacteria. Number 2 is lower human digestive tract disease. So this is a human digestive tract, just a brief review. Okay, you have salivary glands, oral cavity, esophagus, go to the liver, stomach. You have a colon, you have a rectum, small intestinal, appendix, gallbladder. So first of all, we want to mention something. Stomach area is usually, the bacteria is very few because the acid environment, very low acid in the stomach. You know that the pH is about 1 to 2. Stomach acid. Large intestinal areas, there's a lots of bacteria. Small intestinal area, also a lot. When the fecal sample come out of your body, it's a huge bacteria there. Okay, this is a common sense you know. And also in our intestinal areas, there are lots of bacteria, but does not mean they are bad. They are normal bacteria in our body. They could regulate our biological function of the human being, of the animals. And lots of the study has been done that. That's why sometimes we'll recommend you drink some of the yogurt, some of dairy products to keep the balance of your intestinal bacteria. And that is one of your immune system even. So that's what I want to mention, okay? These slides, I already talked about that. Stomach is very few. 100 cells per ml. Small intestinal, little bit large, 10,000 cells per ml. When you move on to the large intestinal, there's a huge population, could be up to 10 to the 12 cells per gram. That's a fecal sample. Majority of them are anaerobic bacteria. So anaerobic compared to aerobic is 1,000 to 1. Fecal sample, your poop sample, 1/3 of them are bacterial cell. This is a lot of people talk about microflora of the intestinal, of the gut area, using molecular methods like 16S RNA, PCR, all those sequencing, that's all come from there. Because it's a big population of the bacteria, it's a target. Okay, so just want to let you know. But you also know it's all normal. Next, we want to mention the first concept. This slide talks about a concept which is we call food intoxication. Versus another concept called food infection. Okay, what's the difference? Food intoxication. You are eating toxin. Food infection, you are eating bacteria. So what are the examples of food intoxication? For example, Staphylococcus aureus, Clostridian, and Bacillus. Those toxins, lots of the time, is exotoxin. And the onset time is very low, about like 6 hours. And after 6 hours, you're going to showing up is like nausea, vomiting, diarrhea, all those things. What is food infection? This is what you heard about the news all the time. There is a bunch of 500 pounds of ground beef contaminated with E. coli 01578H7. There is a hot dog contaminated with Listeria monocytogenes. There is a chicken patties contaminated with Salmonella. That is food infection. So, basically, these are foodborne disease. Cause foodborne disease, such as E. coli 0157H7, Salmonella, for example, and the Listeria species, or Campylobacter, all these foodborne pathogens. Now, another difference is that food intoxication, you need to do a very roughly control. You simply cooking the products will not solve the problem. So, for example, if your food has been contaminated with Clostridium, they may form in the endospore there. You said I'm going to reheat it like 1 hour. Reheat it 30 minutes. May not be killing the toxin. The toxin is still there. It depends what type of the toxin. But the food infection, roughly cooking solve the problem. That's why when you order a steak, they said the internal temperature need to reach 65 degrees Celsius or sometimes 62.5 degrees Celsius. So roughly cooking could solve the food infection problem, but food intoxication may not because the toxin may be already there. It depends really. Okay, so this slide tells you the example is food intoxication. And this slide later on, this here is talk about is food infection. These are all foodborne pathogens. There is a separate class talk about this is food microbiology. So I don't want to go detail all these. We'll mention these later on briefly. Okay. So this is something at the beginning we want to talk about that. Okay. Today's work, basically we want to talk is a big chunk of a group of bacteria is Clostridium. There are a lot we could talk, and each of them we want to talk about these one by one. So let's remove these, make sure it's all blank, then we can roll them. I would go this guy first. This guy first. Okay. So first of all, what we talk about is Clostridium. Clostridium, it is gram-positive, large, very important endospore-forming bacteria. And this is different from what all the bacteria we talked about before. And it is strictly anaerobic. Remember, we talk about the bacteria could be aerobic, strict anaerobic, and right in the middle is facultative. Okay, aerobic bacteria must use oxygen. Anaerobic bacteria cannot grow in the presence of oxygen. Facultative grow both, but better with oxygen. We mentioned about in exam 1 lectures. Now the question in the extra points, why Clostridium is a strict anaerobic bacteria? Because there is no SOD, sulfide dust mutase, nor catalase. Is that right? That's why they cannot survive with oxygen, because oxygen generates during the metabolic process generate intoxication material, they don't have SOD and the catalase to hydrolyze them. Okay, so this is the first thing first, is talk about Clostridium. What are the Clostridian species we want to talk about today? Is Clostridian Difficile, Clostridian botulinum, Clostridian Tetanus, and the last one, Colostridian Perfringes. There is lots of, we can talk about it. So we talk about these one by one. First, Clostridian Difficile. Clostridian Difficile, acronym, just we say, CDIF. Where it comes? This is usually happened in a hospital. Okay, 70-year-old grandma intensively using antibiotics for 7 days a week. What are the antibiotics? Could be amoxicillin. Could be penicillin, okay? Well, amoxicillin is type of penicillin. For 7 days. End up with what? Water diarrhea. Bloody water diarrhea. And if the person to do a very detailed testing, use megacolon, toxic megacolon, and to see in the colon area, there is a very typical membrane colitis. You will see the pseudo membrane there with some of the bloody area. Sometimes it is white, is pus. If it's red, it's bloody. If white, it is pus. That's dead white blood cell. Okay? This is a very simple symptom. Why it will cause this symptom? Because you're using antibiotics too intensively. You're using antibiotics intensively about 7 days. All the normal bacteria has been killed. Remember, normal bacteria in the gut area is killed. There's nothing there. Then this bacteria will be dominant. And this bacteria also has an AB toxin. Same as we talk about the Corynebacteria, diphtheria. A toxin is toxin domain. And B toxin causes bleeding because transmembrane domain. The same thing. So when we do the testing, it's very easy. You just go ahead to do a fluorescence testing of the AB toxin. So you're going to be solved the problem. Okay? Okay, how we going to do the treatment? There is several methods we do. The first one, since we are using the antibiotics is too intensively, we're using penicillin or amoxicillin. So choose another antibiotics. Most of the time here we use vancomycin. But this sounds very ironic. We already using antibiotics for 7 days a week is very intensive because of the problem. Then you said you're going to use another antibiotics. Is that so ironic? Very ironic. Second method, fecal transplant. This is a horrible way to do, but you have to do. Is you're going to transfer the fecal sample from a normal person into your body. Sounds horrible. You're transferring the poop from the other person into your body. It's really nobody likes it. However, sometimes you have to do, because the idea is put normal bacteria back. Okay, that is called fecal transplant. The last one, sometimes the mild symptoms, drink lactic acid, bacteria, fermented products. Let's say yogurt. Because lactic acid bacteria is a beneficial bacteria in the gut area. Kind of they could generate bacteria, could kill it. At the same time, we'll put your normal bacteria back. So that is the idea to do it. Okay. This is a pathogen also happened in the hospital. So that's one of the examples. Nosocomial infection. What the other one we mentioned? Staphylococcus aureus, remember? And sometimes Staphylococcus epidermis, but most of the time it's normal. Okay, so this is something we want to mention, is Colostridian Difficile. Long time ago, we think it's only for the human being, because it's going to be transferred from the fecal sample to the environment or to the food supply chain. So like myself as a food microbiologist, not recently, 5 years ago, people talk about CDIF in the food supply chain. Okay, so we also call it is an emerging foodborne pathogen. Foodborne pathogen. TTHOGEN. Okay, emerging foodborne pathogen. So that is for Clostridian Difficile. It is antibiotic-associated colitis. And you can see a typical here. Okay, now let's go back to talk about Clostridian botulinum. Clostridian botulinum, we have to remove this guy. Botulinum. When we talk about Clostridian botulinum, we want to mention three things. There is three types of the botulinum could happen. Is wound botulinum, food botulinum, and another one infant botulinum. Wound botulinum we go very briefly. This is most likely is some of the drug abused or addictive person easily to have is if you have a wound. Because you are using drug too much, you have the wood, decayed or wood area in your skin. The pathogen will be stay there and penetrated. So this is usually for drug abusive people. Very specific. Not happen too much. Most of the talk about is food botulism. This is related to the canned food. Okay, what is that? So canned food always happen. If you have a can, okay, you're gonna do the tipping later on. What are you gonna put there? Some tomato, some carrot. Of course, you're gonna have like a water. Okay. Sometimes pepper. Long time ago, people only do is cold pack. What means cold pack? You're going to boring 1 to 2 hours, then immerse in cold water. This is not really good. Tomato, carrot, pepper, all those things growing from where? Soil. Is that right? From the soil. When they from the soil, Clostridian botulinum will be dominant there. So once you do the boring to 1 to 2 hours, you're gonna kill most of the vegetative cells. Therefore, the endospore is dominant there, and they're gonna grow. It is a risk. Okay, so cold pack, we shouldn't do it. Then what are we gonna do? We have to do pressure cooker. That's lots of the small industry what they do. Pressure cooker, you need to do 15 psi, 121 degrees Celsius for 15 minutes. This is the idea is to kill endospore. Now the question is become, you are the large industry, you can do pressure cooker. We are a small person, how are we going to do with small processor? How are we going to do that? Then you have to add, is after cold pack or before cold pack, you need to add vinegar concentrate. Or sometimes it's lemon juice. What is the idea for this? To lower down pH below 4.6. Why? Because all the canned food, we call it a low acid food. What is the typical characteristics of low acid foods? Is water activity about 0.85 and the pH is around 4.6. Some textbooks say less than 4.6. In West Virginia, Department of Agriculture said the standard is less than 4.0. We call this a critical control points. We call this a critical operational points. We want to make sure it's safe. So that's why we lower down the pH. Okay, that's the reason. So the canned food, the traditional canned food, you add all those stuff. That's a food botulism. Now, how do you know the botulism is already there growing? The can, you see that it's going to be concave. You see all the liquid comes out. It's become nasty. All the things comes out. Or starts to cranking the cover even. That means all the change the shape. Like this. All the gas bubble comes out. That means it's already contaminated. So lots of the time the sporation is there. Okay, so that's the example. Infant botulism. You only need to remember one sentence. Never feed newborn baby less than 1 year old honey, because otherwise you have floppy baby. Now, why are you going to have a floppy baby? Looks like that. The muscle is completely soft. And the muscle is completely relaxing. Okay, this comes out with something related to the anatomy class a little bit. This is your muscle, let's say. Muscle have contraction like this and the relaxing. Why people can do that? Of course, there's ATPs that would have to hydrolyze. What are the chemicals? Acetylcholine, chlorine, sorry, and glycine. This is for what? Acetylchloride. Muscle relaxing, muscle contraction. What is glycine? Muscle relaxing. Clostridium botulism. And the botulinum, the toxin, is going to be attacking this chemical. And we call these is we call it a neurotransmitter. And if the acetylchloride has been stopped, which means the muscle cannot contract, you're always going to be relaxing like this. That's a floppy baby. How about glycine? If the glycine has been stopped, so muscle cannot relax it, then what's going to happen? You're going to have to contract all the time, is that right? This ends up with the second one, Clostridian tetany. Clostridian tetany, I'll talk usually briefly, because it's not let you relaxing, they're attacking glycine. So the muscle is always contract. Because of the muscle always contract, so what's going to happen? This happens, lockjaw. Locks like this. Why you died? Because the muscle is contracted. Highly contraction. Then where it comes from? Decayed metal. You're walking in the field, walking in the soil, do some of the housework by yourself. If your soil has been decayed with some heavy metal or something, you have a cut on your hands, what you need to do? Go to see a doctor. The doctor will let you do what? Vaccine, DTaP sometimes. It not helps too much, but let you do it. Diphtheria, pertussis, what is there? Clostridian tetany. So remember, Clostridian tetany shouldn't be a big problem in the United States because we have this DTaP vaccine there. That's why all the kids need to have it. That's the most dangerous group where we have to cut in their hands if they're going everywhere to play in the soil, sandy box, or whatever it is. That's why Clostridian tetany is that. Okay? And this is a picture showing you the lockjaw. Cramping, twisting of the skeletal muscles and the tightening of jaw muscle. And diagnose wound infection, muscle stiffness treatments, antibiotic therapy, antitoxin. Okay, and active immune with toxoil. I forgot to mention about this one. Botulinum, when you happen in the United States, usually it is ABE toxin. AB toxin usually for the canned food, E toxin specifically for fish products. So canned fish will have E toxin. And once you have a Clostridium botulinum, what they do? The first thing, they have to call CDC. In the south area, CDC is in Atlanta. And then they have to call the Atlanta airport to ship in antitoxin. The antitoxin usually is made in the horse. So we say it's an anti-equine toxin. Okay? So you must have those. Shipping anti-equine toxin from airport, Atlanta airport, to the places. That's the first thing you need to do. Then you're using antibiotics. That's why canned food is always a hot topic right now, talk about. It's in especially the local community in West Virginia, people like to those farmers markets, made this homemade canned food. It's a small business. So that's why we restrict the pHs lower than 4.0. Okay. Okay, so tetanus. Now tetanus, I always want to talk together with botulism because you can see here, central muscle system, acetylcholine, glycine. Acetylcholine is for muscle contract. So you're attacking acetylcholine is always relaxing. Glycine is for relaxing. So you're attacking relaxing. Glycine, the muscle is always contracted, cannot relax. That's a lockjaw, that's a floppy baby. Okay? So you know this one. Next, Colostridian Perfringes. That's another big one we want to talk. Colostridian Perfringes. What will be the typical symptom for Clostridian Perfringes? Look at that picture. Necrosis and caused by gas gangrene. This is what's going to happen. The tissue is right there. Okay? If you have any of the cracking here, the muscle, this passaging going to come in. Clostridium going to come in. Once they come in, because in this cracking area, this is a favorable anaerobic environment. So this bacteria will going to grow there. In the anaerobic environments, what they do is fermentation. And they're going to be cracking. They have generated gas. How they do the fermentation? They use a muscle sugar. So what happened is the muscle has been broken down into different pieces. And all this area is a gas. These are all the gas there. And finally what happened is the muscle and the tissue, you're supposed to have a blood there. Is that right? You're supposed to have a blood. Now the blood is not going to come in because the gas has been cracking them and have been separating them. So, do they have enough blood to go to the, let's say like a liver area? No, there's not enough. So start to decay all these organs. It is similar to Staphylococcus aureus. Staphylococcus aureus is a toxin there. The toxin low down the air pressure, don't let the blood pressure, don't let the blood go in. Here what happened is, the Clostridian Perfringes will be cracking into your muscle caused by fermentation. Then the gas will be separate all of these muscle. Because there's no blood supply, so that's necrosis what happened. And finally, what you only do is they're going to take out that arm. There's no choice. Oh, okay, because all the tissue and the extra, it's also going to be very invasive. Okay, so that's a gas gangrene. Now, then you're going to ask me, how are we going to prevent this? This is a joke when I talk about when we talk about endospore. Lots of people do. Carrots, okay. Tomato, carrots, and beef stew. And they use so-called sous vide or Swiss vide or sauce vide cooking. When they do like that. Low temperature, long time cooking. Because they're trying to make the food is more tender. What's going to happen? The beef stew, like a bacterial media, beef extract, tomato, carrots, all those things have endospore. So you have a low temperature, not really low, maybe 55 degrees Celsius. You're cooking like 8 hours. Vegetative cells die, the favorable endospore forming. And the sporation is there. The whole box, the whole soup is endospore. So what you have to do, and I said you have to avoid dangerous temperature zone. And what is that? Very easy to understand, 40 to 140 degrees Fahrenheit. If you like me, like degrees Celsius, 5 to 60 degrees Celsius. This is a dangerous zone for temperature of the bacteria. Have to avoid it. So basically, your refrigerator, you have to storage in a cold temperature. Below that, 4.5 degrees Celsius. That's why your home refrigerator around 35, 38 is good. When you do cooking, more than 60 degrees Celsius. 62.5 is good. About 147, 150 degrees Fahrenheit is good. Less than that, you're favorable indoor sport to grow, and you're curing vegetative cells. And that's the only way from the safety standpoint to prevent Clostridian Perfringes. Okay, there's no other way to do. If you have really happened in your body, that will be the end of the results. End of the story. Okay, so that's gas gangrene. Okay, we finished talking about all these, and you can look at the slides. We talk about Clostridian botulinum. This is a pressure cooker. This is lots of them canned food. Lockjaw. And we talk about the principles behind it. And we talk about Catalase Difficile. So here we're going to want to talk about is foodborne and waterborne infection. And I just mentioned this is called a food infection. So we want to mention a little bit briefly. First of all, Salmonella typhimurium and Salmonella typhi are different. And you should know. And I want to tell you one thing, this textbook right there is wrong. Salmonella is italic, but typhimurium, typhi, or typhimurium should be normal because it is a subspecies name.